There are well-known mechanisms involving T lymphocytes in the pathology of type 1 diabetes, but B lymphocytes also have a role. This can be demonstrated in a strain of mutant mouse which develops a type of diabetes similar to that of humans, which are protected from diabetes by mutations which cause the lack of the Ig mu chain and the lack of B cells. Also, these mice are protected from diabetes if given anti-IgM antibodies, the antibody expressed on the surface of B lymphocytes. While T lymphocytes are clearly involved, B lymphocytes are important antigen-presenting cells which help to stimulate naive T helper cells. B lymphocytes should not be able to generate antibodies against self, so they should not be able to express antibodies that would bind to insulin and recognize it as foreign or to any other protein produced by the beta cells of the pancreas. There are numerous mechanisms in both central tolerance and peripheral tolerance which should stop this. But nevertheless, B lymphocytes are produced which make self antigens, including those against insulin. And these can be devastating to the beta cells because when antibodies bind to cells, this can attract the complement proteins out of the blood, which generate a pore on the cell membrane and destroying it. Also, when antibodies bind to antigens in a process known as opsonization, that attracts cells which can perform phagocytosis, such as this macrophage. The macrophage can then present antigens from the ingested beta cell, which can then further stimulate cytotoxic T lymphocytes. While the development of antibodies against insulin are a major early step in the development of many cases of type 1 diabetes, they are not an essential step. In this first young patient, this individual does develop antibodies against insulin and develops insulitis, the inflammation of the islets of Langerhans, which then attract both B and T lymphocytes to the islets. However, the disorder need not progress any further. And if these are the only autoantibodies that this individual makes, they will probably not develop diabetes. Most of those who do not develop additional antibodies, only one set of anti-self antibodies, will not develop diabetes. The second patient also develops antibodies against insulin and insulitis, but it progresses. And these antibodies against insulin are commonly seen in those who develop childhood diabetes, more likely than not. When this individual then makes a transition to preclinical diabetes, they develop antibodies against 2 or more beta cell proteins, and the number are given here. This can then cause the individual to develop type 1 diabetes in childhood, the vast majority of which develop antibodies against insulin before they develop full-blown diabetes. This last individual developed diabetes later in life. And like most of those who develop type 1 diabetes after age 15, there were no measurable anti-insulin antibodies. Thus, these antibodies are commonly one of the first symptoms in type 1 diabetes, but many cases occur without them.